Hello and welcome everyone in another episode of Researchers Celebrity powered by Empowering Science Foundation. Today we have Dr. Jasbeer with us as our researcher celebrity. A brief about Dr. Jasbeer. He has done his bachelor's in biological sciences, master's in biotechnology and PhD in molecular biology. All the education, all the degrees were earned at Maharshi Dayanand University, Rohtak. Then he joined postdoc at Jawaharlal Nehru University and at ICGB New Delhi, followed which he joined Dr. Lal Path Labs in molecular diagnostic groups, and currently he's head of molecular diagnostics at iGenetic Diagnostics, affiliated with Manipal Hospitals. With this brief introduction, I would like to welcome Dr. Jasbeer at the platform and saying thank you very much for taking time out of your busy schedule.
Thank you. Thank you, Raja, for inviting me on this platform. So I really appreciate that the, the way you have started that to discuss about that what the researchers the problems while doing their PhDs and other research. So this is actually a good platform where we can share our experiences and to our following the upcoming researchers, and we can add up with the things to which will help them with coping with some issues that we have faced at that during our time. So if I
So we can start the conversation with that's how we always start our conversation with the standard question that how and when you decided that you want to become a researcher.
if I talk about that in that sense, what to decide that when I'm going to be a researcher was typically when if we go back to our childhood. In that sense, when we since my childhood, I was that much fascinated by the intricate working of the biological system. That's typically the totipotency of the stem cell, right? So that that is a big term, that but actually what we have in the layman, that we have when I saw a lizard having is surviving after cutting off its tail, and it's regenerating again. That was very much fascinating when I saw on that crawling on that on the surface, and decide that why this lizard is having very different kind of color than this part of the body. So that was the things when my curiosity started on that. So, with due course of times, I was much fascinated that for the biological sciences, I was interested since when 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 I was in ninth class, and then the science was much more what to say that is was more attractive to me rather than the other science and mathematics, basically, rather than the other subject. So it when I was in 11th, I there was some problem in my domestic problem. There was some problem. I lost my mother at that time when I was 11, and I had to come back from the Rohtak to my hometown, and there I joined in a college where there was no science in that, and so I took geography and mathematics in that as in 11 subject, and I topped the college that year. But that was really not a cup of tea for me, my cup of tea. So that's why I I started again 11th with the science studies. So that was the thing that was keeping me that I wanted to do the things in science only. So I'm not want don't want to do the things in any other field. So that was the thing. And post uh, when I was in BSc second year or BSc second year. So when I met my one of my there was a new teacher that has come from Punjab University, and she was just teaching. Dr. Kakul Bulia was there, and she was just teaching us the plant biotechnology system. And in um, that course, I was that was very much fascinating for us, because that was regenerating of the plant from a single cell, or regenerating a plant from just cutting of the stem or cutting of the root, and artificial rooting, and those things were very much fascinating that time. So, and it was a new field for at that time, and it was a lot of research was going on. And then I cleared my uh, 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 entrance examination in Andhra Institute, and I choose biotechnology as my subject, so that I can pursue my dream, so that I can fulfill my dream of that I wanted to do something uh, uh, the biotechnology part. So that's the thing, that but biotechnology is a multi disciplinary subject. So that was good, and in MS in masters I topped the university again, and then I decided to go with that uh, in the um I clear my CSIR and ICMR fellowship in, and then I got an option for from AIMS, where the Dr. Sunita Sena was there, and she offered me the project in the tuberculosis, so that I should work in tuberculosis. But that I wasn't much interested in that. And later, again, I found that I got a proposal or request from the National Institute of Oceanography, Dr. Usha Goswami, where she was planning to to start a project of uh, next gen sequencing, for genome sequencing of the Parnavides. Parnavides is a marine mollusk, and it is having some medicinal, and it's delicious for it's very much commonly eaten in in the Asian countries, and it has some medicinal properties for patients who have arthritis. So they wanted to explore the genes and other things, that other novel genes, to find out that they got a project of for next gen sequencing of the genome sequencing of this Parnavides. So I was the first person who has joined with them, and she was very happy that someone from that so far from Delhi, and he he's enthusiastic in doing the things, and we started work from there. And that was collaborated with the National Chemical Laboratory Pune and with Dr. Mohammad Islam Khan. So we had a close collaborations for the uh, sequencing part of the sequencing, and there are something which products that were beneficial for the human being. So to extract those kind of product from these things was the part of um, Dr. M.I. Khan. So we shared that. So I partly I was working there with Dr. Khan, and fetching out the DNA and the protein, and in the reverse transcriptomics, and then it's to fetch out the particular genes on that. And meanwhile, those who tag those genes, so the other 10 guys were there, 10 other fellow members were there, which are working in different part of the things. And I spent around 1.5 years, more than 1.5 year, I guess. And there was one issue at my home, and my father got paralyzed and some brain hemorrhage, and I had to come back to place, because no one was there to take care of him. So that was a big challenge for me. So initially, I approached AIMS Delhi, and where the Dr. Hassan Das has offered me, I joined, offered me a joining letter to work with him. That was fine, but again, after a week, again had the same problem. This time, the other uh, part of the body was affected of my father. So I decided to stay at home, near home, so that I can take care of him while doing my uh, research. So I joined in Maharishi University, Rohtak, in the biotechnology department with Dr. Jaiwal and Dr. Aarti Singh. So, and there, that was the thing that I started working with the plant, regeneration of the plant, and testing the rotavirus protein gene in that. So that was the things. That was the main thing that I did in my PhD, and this doctor research that uh, was the thing that provided me with a solid foundation on the molecular biology that allowing me to delve deep into the mechanism, disease, and genetic mutation. So post PhD again had the privilege to honing my skills through postdoctoral fellowship at
So Dr.
um,
Jasbeer, here we'll take a moment to discuss
yeah.
a little bit about the PhD. So what you have shown and shared right now here, the challenges which came, and this is for all the audience here that Dr. Jasbeer is perfect example of any researcher, that how researchers never gives up. He had faced issues starting from not go having the school where he can pursue science. So he joined arts program, excelled it, still was not content. So went back, started everything again in science. And as we see today, he's leading the diagnostic department. But what is in the foundation of it is that how at different stages of his life, he has faced family emergencies where he has to go back, take care of the family, which is our support system. And for most of us, family and friends are the support system, which always take us out of the deep ditches in our life whenever we are feeling low or so not content with the life. But here I would like to talk about two, three different things for all the researchers who are either pursuing PhD and facing issues, or, you know, are planning to consider PhD as an option or the only option for their life. So what we have just heard with Dr. Jasbeer, that how he joined National Institute of Oceanography, spent almost 2 years there, then joined AIMS, spent a couple of weeks there. Finally, at MD University, he completed his PhD using all the experiences he gained in at different places and obviously the expertise. But what he already has established with the other two places was a reputation, a networking, a one-to-one known person, where they know him as a researcher, they know him as a person, they know him as a human being who respects the family, that he's ready to sacrifice, or not even thinking about that he's sacrificing his career because he has to take care of the family. These are the things which sometimes we all face issues. Sometimes we chose career, sometimes we choose family, but this is so individualistic that every person is in a right place to decide what they want to do and how they prioritize it. Now, coming back to Dr. Jasbeer, when at the end of the day, when you earned your PhD, what is missing is the part of your research which you done at your MD University, along with taking care of your family. How much challenging was that? And then based on your experience, what you can suggest to the new generation of researchers, that if someone is facing the similar challenges, how they can overcome?
Um, rather, the first and the biggest challenge that I faced when I joined Dr. Jaywal at Marshall was the availability of seat that I had to register for PhD. So there was no seat actually. Then I waited for 2 years to get it get my turn registered for the PhD. And there were the things that if you register post registration with the PhD, you have to spend at least 3 years for your to get a degree and for things to be completed in that sense. There were things that was the custom that was made made for that every researchers in that lab. So those were the things. So initially, I waited for 2 years to get it registered for my PhD, and later on that there were the things because the topic I chosen choose was the, a plant that was uh, this was the which was that that research was a pioneering subject, and no one has done the work on that plant there, and I chose that plant. And actually, my my mentor, my guide was also were also not aware of those the deep issues that I was going to face in the future. So those was the very much. This was very much a new start, a, a diving in dark ocean. So it was like that. So it was challenge for me. So but I started working my own, setting up an medium and culturing the part, and then regenerating the cells, regenerating the plants, and then the introduction of that. Another thing was that there was because the lab that I worked with was not having any expertise with in relation to the any kind of viral or bacterial pathogenic diseases. So, but that was my own aim that I had to transfer those kind of things, uh, uh, make the plant genetically modified with that some kind of protein genes that are beneficial for which are immunogenic to some kind of particular disease. For that, I I I took help from the DRD Gwalior, Dr. Goyal, and another institute there where I worked and took help from uh, HAU, Hisar. So Haryana Agriculture University Hisar, Darshana was there. So she helped me out with these kind of strains that they were having, and how to culture those strains, and how to keep them sustained on that different media. And those were the things. This is a part of the research that I chosen, and though my mentor was not having any kind of experience or any kind of guidance, that I uh, I was not getting much out of that. So that was the thing, everything I had to do my own. And that time, resources were limited in 2004. Uh, that was advancement was not that there. So we have to wait for liquid nitrogen that we have to collect it from 30 kilometers away from my city, and we had to go by bike and to fill a cylinder with that. And working and taking in the state university was the biggest, uh, it is the biggest challenge that you have to deal with the financially. So for the first 1.5 year, I didn't get my fellowship from CSIR. So there was some issue with that transferring of my fellowship from NIO to MDU, and so some and the state universities has their own guidelines and kind of protocols that how they work. That is that I'm not going to tell about that person, but that is a quite uh, very difficult to handle the situations, handle the things in with the people who are working within the state university. So the procedure is quite different than if you work in a good institute like uh, ICGV or any kind of CSIR or ICMR institute. So that is a very different thing. So there, those were the major challenges that for small things we have to wait for that. And so for primers, then we are waiting for at least we are spending at least a 1 month so get it approved from the for financially so that then we can put an order for those primers. So for in between that, if I remember a couple of times that I went for PCR machines that we bought in our project. So I personally went to to the airport for collect for collection of that PCR machines to get it cleared from the custom, and that those I think these were small things, but that was impacting on our the, wasting our time in procuring the things. That was the biggest. I think these were the biggest hurdles that the thing. So these kind of small things that I could recall. So these are the major things I could recall, but small things I am not able to don't want to discuss. So that's fine. That is every researchers uh, faces, but I never recommending that to
This is, I think there is a little bit of technical glitch, but here I would like
do the
to,
research from a kind of resources. Right?
yeah, I would like to summarize the challenges which Dr. Jasbeer faced and also would tell to all the PhD students who are currently registered or are planning to register for their PhDs that sometimes you go by the name of the group, the reputation of a mentor, and there is a wait list already in the lab or in the research group, as Dr. Jasbir also faced, and then he had to wait for 2 years just for the registration. So if you really want to join a lab where there is a wait list in there, the way you can overcome it is if you have a clear conversation with your advisor that, okay, I can wait for 2 years, but then the minimum time is for submitting a thesis is 2 years also. So if we can work on a project which is complete within 4 years so that you can still have your degree in total 4 years, even if it is the registration which says that you have completed it in 2 years. But it says that you have already put all the efforts which it needs to be required to accomplish your project goals. So that is one way you can overcome it instead of working or deciding your project only after your registration, which is like again adding 2 more years or whatever the number the wait list is there to your research. The second point was in-depth knowledge of your proposed topic of your research. So you have to make sure that either the advisor you are going to join PhD with has expertise in it, or if not, then at the minimum, he has or she has the collaborations with the person of the expertise required for successful accomplishment of your research goal for your PhD. A very important point about the resources. Either it is infrastructure or it is about the administrative support which you get from your affiliated institute or university. We all know that in India, sometimes it is because of the financial year changes, sometimes because of some other changes. What we cannot take care of is how the government works, but what we can take care of is how we can make our administrative staff understand that what we are doing, because the people who are sitting in administrative offices have no ideas about your transformations, your transgenics, your PCRs, or any other technique which you are doing in your labs. Neither they are known, or most of them have no clue about the biological functions, the why you need as small as a plastic cup for your experiments, which they think is good for popcorn cups or anything similar like that. So how we can overcome this is if we can have a science day or a science week once in a year, so that all the administrative staff can be introduced to all the research groups and show them where you are actually utilizing the things where they are questioning that why do you need so much of paper towel, or why do you need so much of plastic ware, which is like or detergent, washing detergent or something. Because when you tell them that you are using the things for those purposes, and now they understand, next time if you are going there with your approval or your application that okay you need this, so they already know that why you are utilizing this, and then they are more understanding and sympathetic towards you to enhance the process. So now, even if you are saving 1 week or a month, that is going to help you big time in your research. So now, Dr. Jasbeer, now we'll talk about the post PhD journey of yours. That with all the difficulties, with all the challenges, you successfully accomplished the attainment of the PhD degree. From there, when you decided that you want to still remain in the research, were there any roadmap you already laid for yourself that this is what you are going to do, or you left it open for the fate and enjoyed the research as it come to your way?
No, I think this post PhD experiences were very much positive for me, and those were the years that uh, that that gives me privilege of on skills. And um, uh, I firstly invited by Dr. Neera Bhalla Sarin that there was one project, and I applied for that. There was project, and that was a limited type project, but that she told, and but she preferred me over the other candidates, and she that was a good opportunity for me to join with the Ranira Valasari, the Jordan School of Life Sciences. So there were the research that I was involved with the pathogen related 10 protein gene that to be which was to be expressed in the Arachis hypogea and groundnut. And the I did my best out of that, and I could there I could share. That was a multi-institutional product, and I used to train other people as well for that who are in that same project, and for the different techniques that we used to get the uh, faster transformation of that particular uh, plant. So those were the things that I was appreciated by that team, and I think it's a good time, a really a good time that I enjoyed there with doing search part of the team with that Jane other things. So by that time, so as the project was over, so it was a limited time project. So uh, I I got an opportunity with in the synthetic biology group of ICG, International Center for Engineering and Biotech, and there it was a good opportunity for me. That was an entirely new thing, uh, but it was basically the doing the thing with that DNA and to 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 create some transformation vectors and other thing which was the targeting was kind of uh, marine algae, and this was a well. there I brush up my skills, and I got expertise that in piece, real-time PCR and next-gen sequencing and designing development of the complex math. So this kind of things that I enjoyed in ICGB engine. And before I have completed the things there, so there was a it wasn't just a project meet, and I met with the people of Alpat Labs, and said that why don't you join us but we need a good uh, professionals for our molecular tests that we are developing for NGS as well as field and things there. Then I thought that it is a good opportunity, though uh, that there I'll be. I was involved in a project of to create a test for tuberculosis, and that was Dr. R.K. Bhatnagar at School of Biotechnology in JN and Dr. Lal Prasad. These were the two institutional projects was there, and that we have to create a kit for tuberculosis. So that was a big thing, and it was quite interesting. For that was that I can take part in that developing of these kits, and uh, which will be helpful for testing the tuberculosis patient. And tuberculosis is the biggest threat in India in developing countries. So that was clear that the music credit and doing I had done entirely that all tested for 3,000 more than 3,000 of the samples, and that we tested with the gene expert culture and our own generate own created parameters and props. So in that case that we uh, we publish a paper on that. So it was a good time. That was a good thing that I came for. That was lucrative for me. So that was the thing that I started with, and later on that PI was. This was actually a professional journey. So it was a significant turn for me. And because I automatically it was not possible to automatically transition into that thing, but it took time to transition into the corporate world. For 7 years, I was there with Dr. Lal Path Lab, so doing as a senior research scientist and doing the development of assays, molecular assays, whether it was for infectious disease or in oncology segment. So those were the things. And apart from that, I was also chairing another department of transplant immunology. That was another fascinating thing that I wanted to do. So so NGS, I took training on NGS for HLA typing and started working. That was the thing with there. I was shared between two departments. One was the molecular diagnostics, another was the HLA typing of and immunohistochemistry. So these were the two departments that were was sandwiched, and it was great. I was clearly enjoying. I was completely enjoying that my tenure up there. So at um, after a while, after some years, that I thought I have enough expertise now in the molecular diagnostics fields, and I should do another um, uh, company or job or other areas of the field. So I got an opportunity that with the iGenetic, which was just started. It was the startup in 2 years. 2 years it was 2 years it has completed. So those at the time, so was I joined in October 2019 with the iGenetic, and later on with this diagnostic uh, form was taken over by the Manipal hospital. So uh, that was a lot to learn about that, okay? Because in this laboratory, we started a multiple kind of uh, a kind of testing that takes multiple tests on a one from one single sample. This testing would be helping to the patients who are very critical on that bed, or they are on very serious conditions. So kind of tests that we wanted to we started were the septicemia, sepsis test, where that's utilizing taking the only utilizing only one sample at a time and doing the 32 different tests on PCR based tests assays on that. So that was the 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 idea was that they were using a very little amount of samples, and we can do a lot of many tests for the patients, and that will help escalating their antibiotics or antiviral, antifungal treatment. So that can be escalated within a day, so that it can save the time of the patient. It can save the life of the patient. So it saves the time of the to stay long in the hospital, and it will also help the doctor to keep his reputations in that. So those were the things that we started doing this single sample multiple tests that we called them as infection panels, and these were the things that we started doing. And then we encountered COVID uh, pandemic and that. So
Yeah.
COVID-19 pandemic was an outbreak in 2020, and it changed entirely. The system was changed entirely. The the objectives didn't change.
Yeah.
So everything was shifted to the COVID-19 testing and all these things. So those were the things that it was actually because this is a corporate world, and it was a professional achievement that the leading a team of um, a good technical staff, which they're almost all of them are masters, or they have some research experience as well. So this this in the past, in those 3 years, that was the revenue of this company sells to 20 sells to 50% in these 3 years. So
Mhm.
that was a good. I I was just it was just and this a pleasing moment, and where I can where I could demonstrate my ability to blend the
Yes.
scientific acumen with that strategic business development. Those were the things that which uh, I think were the good part of my uh,
Absolutely. And
this
this is not
entire
only,
business journey.
yeah, this is not
Yeah.
only your good part. I believe that this is accomplishment for any researcher in the world, like increasing the revenues by 250% and also being part of that. So, keeping the time limit in mind, Dr. Jasbir, the last question for this conversation will be like, in you so you have perfectly balanced your life till now in academic research as well as in industry research. At any point of time, have you ever thought of quitting research either when you were in academia or in industry? And if the question is if the answer to this is no, then I would like to ask a quick comment from your side that what you suggest for the researchers who are actually having the thoughts of quitting the research either in academia or in industry now?
So my definitely my answer is no. That I never thought of quitting the research or I, in that. whether I am in service or at that time of my academic research. So I never thought of that thing. So and it was not in my, and I never thought of that because I like to do that thing. So whether it was my that all depends upon your passion and it all depends on satisfaction. So how it is going to affect that, how it is going to please your inner self. So that is the thing is. So I'm always maintaining an unwavering positive outlook that I'm working. Initially, I started working uh, in an academic part, that I'm doing something new, which is going to be helpful for a community, for an all people of the India, for people of the world. And here that serving, I'm serving in this industry, that I'm working for a patient. I'm not working for any kind of organization. Getting the revenue is a different thing, but my main motto is I'm serving for a patient. I'm working for a patient because I want to do. This is my motto, that I want to help the patient, that to uh, get out of the uh, uh, hospital and get back home on his own legs. So those are the things that uh, I always never that never led me to come out of that.
Nice, nice. So with this, to all the audience, that Dr. Jasbeer shared the journey, the enthusiasm with you all, and either it is fundamental research or it is translational research, until and unless you are enjoying it, I think whatever you are doing is helping the humankind in whole. And always make sure that if research excites you, keep enjoying that. If you want to write to Dr. Jasbeer for guidance, mentorship programs, you can contact directly him. Or if you want Empowering Sense Foundation to help you, we are there to connect you with our researcher celebrities. We have mentor-mentee program, one-to-one mentor-mentee program, where you can get advice from them, mentorship from them for the challenges you are facing or how to make life easy. Either it is about the career transition or it is about your current experiments, whatever it is, we are there to help you. So stay tuned for other episodes of Researcher Celebrity. And with this, we'll say thank you very much, Dr. Jasbeer, for sharing the journey and igniting new generation of researchers for their to pursue their passion.
Thank you, Dr. Kushwa, that inviting me to on this platform. And this was absolutely very much exciting to go back to my yeah, that time and then the recall those things. It was good moments, and that I enjoyed and reliving those things. Thank you very much.
It was a pleasure having you as our researcher celebrity. Thank you very much, Dr. Jaspreet.